This is then the flyer to our workshop, and which is organized by Anton Arkhipov and myself. And this is actually the second in a row of workshops where we talk about this like multi-purpose models. And what is really a multi-purpose model? Well, if you look at other fields of science, for example, weather forecasting or fluid dynamics or material science, we now have modeling tools or modeling schemas, or in some sense multi-purpose modeling schemes that can be used to compute many things. Meaning, like you have like good weather forecasting programs can predict all kinds of weather and so on. So, and in material science, for example, we now have these very efficient theories or methods for computing optical and electronic states of all kinds of materials, essentially solving the Schrödinger equation. So what is the situation in neuroscience, and in particular when it comes to network neuroscience? If you look at the studies of cortex, I mean, until now, the systems neuroscience have largely focused on neural representations. How are different features of the world represented in the brain? And the prime example is sort of like these receptive fields in the primary visual cortex, where they have found cells with all kinds of different response properties. I mean, orientation selective cells, direction selective cells, and so on. And this work of mapping these receptive fields has been going on, I would say, since the 60s, 50s, 60s, not only restricted to the visual neuroscience. But what is the status here? Well, there was this mini symposium at the Society for Neuroscience, I think in 2005 or something, 2004, where like a bunch of leading visual neuroscientists discussed, do we know what the early visual system does? And this was sort of then after like almost 50 years of like the mapping out neural representation. And they said, the first line of the abstract said, we can claim that we know what the visual system does once we can predict neural responses to arbitrary stimuli, including those seen in nature. And this was certainly not the case then, and it's also not the case today. We don't really have models that can predict all these things, at least simultaneously. So what they really were asking for was this, what's called a multi-purpose model, a model that can sort of predict many different things simultaneously. So what is the status of the present modeling of primary visual cortex? And the primary visual cortex is sort of where I would say that where the modeling has, at least mechanistic modeling has sort of, has had most success. But still the typical models are what we call low dimensional models, maybe one or two populations of neurons, excitatory neurons, inhibitory neurons. And they're typically designed to predict a particular thing like orientation selectivity or direction selectivity of these cells. So they are in some sense single purpose. Then we also have these other models which has been designed much more bottom up. And one example is this CAT V1 column model, 80,000 neurons from Potjans and Diesmann, about the one, like representing about this 1 square millimeter of the CAT visual cortex. And then recently, these models that came out from the Allen Brain Institute in the group of Anton here, where I would say like the state of the art in terms of like bottom up detailed models, so they had like 230,000 neurons was published last year in Billy et al. And it comes in two versions, both the sort of like the biophysical detailed version and the point neuron model version. But at the moment, there's no, I would say like there's very weak connections between these very complicated high dimensional models. High dimensional mean that is like many differential equations and obviously also many parameters need to describe these differential equations, and this low dimensional model that are sort of tuned to do particular things. So what would a multi-purpose model for primary visual cortex look like? Well, it would be models that simultaneously can predict experimental data for different visual stimuli, for different measurement types, for different brain states. And also then, and these multi-purpose models must likely be high dimensional. I mean, like real brains. I mean, typically if you come from physics like me, you like low dimensional models with few parameters maybe, so that you can sort of find an optimal parameter set that sort of explains some feature you're interested in. But these multi-purpose models must likely be high dimensional. So it must be typically at these like more detailed levels, like in the like network models with many neurons. And you also want them to, in principle then, or not only in principle, also eventually to be able to be reducible to several of these single purpose low dimensional models that explain different features. We wrote a paper a couple of years ago where we sort of outlined this, I would say, like these ambitions for a multi-purpose modeling and the development of that, and the critical case for using, well, the critical importance of having these large-scale brain simulations to explore or to develop and test such models. And we also then, in collaboration with our groups in Norway and the groups of a group of Anton at the Allen Institute of Brain Science, we have then started working maybe towards sort of like making such multi-purpose models by expanding the modeling work that was done in making this V1 model, which was then tuned to predict some kind of spiking behavior or some aspect of the spiking behavior. But now what we bring to this now is that we also can compute the local field potential that the same model would produce. So that put more constraints on the model. So we hope then we'll be able to make it, well, put more constraint and make it possible to identify more of the parameters in this model. And then, of course, also taking advantage of this fantastic data that also has been recorded at the Allen Institute. So Anton will talk about this, both the model and also all the different resources that is provided by the Allen Institute, which is very important for, at least for this particular behavior. So what we did then eventually, if you sort of dream of what the final theory, whatever, or like a better theory should look like, it would be like a set of models at different levels of detail, which are then internally consistent. Maybe you can sort of derive or reduce under certain approximations these high level models from the lower level models. And then they should also be able to predict all kinds of measurement modalities, not only spikes, but LFP and EEG or what have you. And then you have this goal of trying to iterate between these candidate models and the experimental data and go back and forth so that you are able to identify models or this class of models that is best able to predict the experimental data that you have, well, at a certain moment in time. So there are many challenges to be discussed at the workshop, and in particular this, how do you fit these high dimensional models to data? Because low dimensional models with a few parameters, that we know how to do, but these are high dimensional models, many parameters and many differential equations describing the model, and also then complicate the data. So there's one question, sort of what is the best way to, well, what are the methods we have? I mean, basic techniques versus more like what we call standard optimization techniques, multi-objective optimization. How do we, if there are like, well, what aspect, if there are more than one aspect of a measured data set that you want to model at the same time, you maybe have several objectives, and you have maybe more data types. So how do you deal with fitting a model to different types of data at the same time? Spikes, LFP, it's like 2-photon calcium imaging data. And then also development of test suites and benchmarking data. And then also all kinds of optimization involves speeding up, involves a lot of model evaluations. So are there tricks we can use to speed that up? Do sort of like what we call meta modeling, so that you can sort of essentially get faster model evaluations. And then there's a whole different aspect. I mean, now from machine learning, we know that there's like possible to learn models that just has some kind of learning rules, so that the model sort of in some sense self-tunes to predict particular experimental data. So this is also an aspect then that will come more talks about this tomorrow, where sort of like insights from different types of artificial neural networks, convolutional neural networks, recurrent neural networks, how that approach can help us to identify this particular model. Because the good thing is that you have learning rules, so it's easier to identify, or you have like a procedure for like finding the right parameters earlier. The downside is that these models are typically the neural nodes, the dynamics for each neural node in network is simplified or doesn't correspond to the biological reality. And then there's also other things we'll discuss, like insights from other subfields in computational biology and also other things. So that's essentially what I plan to say.